everyone for coming. I will share my screen here. Let's see. All right. Great. Stephanie, that's a hard act to follow, but I'll try. So what I'm going to talk to you today about is a project that has been the result of a really long-standing collaboration between my lab and Dr. Claire Chegnier's lab. We have a very tight collaboration here at Cincinnati Children's Hospital. And this project was really started by this graduate student in my lab named Jana Rayner, and then followed up with her cohort, Maha Almanan, who kind of picked up the project after Jana initiated it. And the project centers on aging and immune suppression. And so one thing to notice in this graph, animated graph from the World Health Organization, is that people are living longer and they're accumulating. And so by 2050, in many, many countries across the world, 30% or more of the population is going to be over age 60. And so that's basically a doubling in the next 30 years. And so this increase in the age population will be a dramatic burden on society and healthcare costs. So as everyone knows, there is a lot of outcomes with aging that range from disability, frailty, age-related diseases, out to longevity and centenarians and long-term, healthy aging. And the balance between, disability and frailty and longevity really seems to center around this concept of inflammaging. So the more inflammation you have, ongoing inflammation, worse age-related diseases are. And the more anti-inflammatory capability you have, that is associated with longevity, right? And I'm talking about the whole person kind of level, right? In terms of the immune system, it's been studied for many, many years, and it's well known that there are kind of hardwired problems with the aging immune system. So if we look at T cells, there's obviously thymic involution and loss of naive T cells, which limits your naive T cell repertoire. There are well-known defects in the formation of T cell synapses, and of course then early defects in TCR signaling have been very well documented. There's also oligoclonal memory CD8 T cell expansion, which also limits the memory repertoire. It can constrain and eke out other memory cells from the repertoire. On the B cell side, older humans have fewer naive B cells, and again, the repertoire diversity is compressed. They have a diminished ability to undergo class switch recombination and lower expression of AID. And of course, then there's defective T cell dependent B cell responses in the elderly. An accumulation of TBET positive age-associated B cells, which may also contribute to autoimmunity, right? What Claire and I have been noticing over the past several years, along with a few other groups, is that aging results in an accumulation of FOXP3 positive regulatory T cells. And they accumulate to the point where if you take a 1.5 to a 2-year-old mouse and you look in the spleen, you'll find that 30 to 40% of the CD4 cells are FOXP3 positive. And we published this in multiple papers along with other groups. And so we wondered if this accumulation of immune suppressive cells was contributing to age-related immune dysfunction. And this is really kind of a different twist on the topic, because it suggests that while there may be intrinsic defects in cells, there may also be a contribution of active suppression of the immune system in aged individuals. And so for these experiments, I thought I'd go through a little piece of definition For the mouse experiments that I'm going to talk about, our young animals were between 1.5 and 4 months of age. The middle-aged animals are between 11 and 16 months of age, and then the old are above 16 months. And this is pretty closely associated with humans that are in the young under 30 years of age and the old over 65 years of age. And that's a close, I would say, a close approximation. And so one of the things that's well known, as I kind of referred to before, is inflammaging. And one of the hallmark factors of inflammaging is IL-6. And we, along with many, many other labs, we are not the first to talk about this by far. This has been known for probably 40 years, that with age, serum levels of IL-6 are increased in both mice and humans. But we had just seen this as well. And the way that we are measuring cytokines in the mice, I think, is particularly instructive. So we have been using this assay that was developed here in Cincinnati by a guy named Fred Finkelman. And what Fred discovered was that you can inject an antibody, a biotinylated antibody against a cytokine, and it works for some cytokines, not for others. But we have one for IL-6. We also have one for IL-10. And what you do is you take that biotinylated antibody and you inject it into the animals, and then the next morning you come out and you basically do a capture ELISA for IL-6 or IL-10 for your cytokine, and then you come back and you measure the biotinylated antibody that's bound to the cytokine. And what it gives you is, rather than giving you a very close snapshot in time, if you just measure cytokines in the serum, which are often very unreliable, this gives you a steady state level of a cytokine over a 12-hour period. And so it's a much more reliable measurement of cytokines in animals, and allows you to directly compare animals. And so here, we use that approach and found that, like many had reported, old mice have 2 to 3 times as much IL-6 in their serum as young animals. But as you all know, that anytime you get an inflammatory response, the animal mounts a massive anti-inflammatory response. In fact, most times when you sample a piece of tissue that's inflamed, and then you do an RNA-seq or some type of genomics analysis, and you expect to find all these markers of inflammation, typically what you actually find is the anti-inflammatory response. And that's pretty typical. So we wondered, what's happening in the context of inflammation with age to the anti-inflammatory response. And so, on a whim one day, I said to Jana, why don't you just measure IL-10 and see what you find? And she came back and she said, wow, she said, it looks just like IL-6. It's actually increased 2 to 3 times in the serum. And we have this really unique exchange program at Cincinnati Children's, where we partner with a university in Germany, where we have an international research training grant with the University of Lübeck, where students in Germany are mandated to come to Cincinnati to spend 6 months to a year in our labs training. And then our students have the opportunity to go to Germany and study in their labs for 6 months. And so Iridi here was in the lab, and I said, hey, Iridi, why don't you take apart the mouse, and why don't you let me know what tissues are responsible for this increase in IL-10, because IL-10 can be made by a number of different cell types. And so when Iridi did that, she found that the majority of the IL-10 was really coming from the secondary lymphoid organs. And in old mice, this epididymal tissue, this white adipose tissue, really becomes like a lymphoid organ that's filled with immune cells. And so that led us down this path of, this is seems to be coming from immune compartments. And so we wondered what were the cellular sources of IL-10 with age. And so we had these mice that were actually made here in Cincinnati by Chris Karp. And these are called vertex mice. So these mice have an IRES GFP cassette knocked into the IL-10 locus. And so any cell that's actively transcribing IL-10 will be GFP positive. And so Maha just took the mice and stained them. She just sacrificed a bunch of young and old animals and just stain them for markers that identify B cells, such as CD19, identify T cells, TCR beta, and then looking in CD8s and CD4s, and then non-B, non-T would be the CD19, TCR beta double negatives, right? And this was directly ex vivo with no stimulation at all. Right? So we just take the cells out, run them on flow immediately. And of course, we find in the in the young animals a lot of different cell types. B cells and non-B, non-T and CD8s and CD4s make a low level of IL-10, around 1% of the cells, right? But when we looked in the old animals, virtually all the populations increased a little bit with IL-10 production, but by far, there was a massive increase in CD4 cells that were making IL-10. And this is just shown in the graph here. Around 5 to 10% of the CD4s were making IL-10. And if you'll notice, if you've got a really clean eye, you can see that the amount per cell was doubled, if not tripled, compared to some of the other cell types in the amount of IL-10 produced per cell. So CD4s, not only are more of them making IL-10, but the amount that they're making per cell is increased. And so we thought, well, this is going to be a waste of time, because we know that Tregs are increased, and we know that Tregs they make IL-10. So, but I said to Jen, I said, just humor me and take the mice and sort the Tregs and then sort the memory cells and the naive cells. So she got a hold of FOXP3 DTR GFP mice, and she isolated to high purity the Tregs and the memory cells and the naive cells. And then she just took them and stimulated them with PMI-adenomycin and said, okay, we're going to measure IL-10 in the supernatant. And this is where the story got kind of interesting for us, was that we found that, as we expected, IL-10 coming from FOXP3 positive cells was increased significantly, but it was dwarfed by this massive increase in IL-10 coming from the memory cells. That surprised us quite a bit. And so I said to Maha, I said, okay, you have to show me that this is really important for the increased serum levels of IL-10. And she did the following experiment. She took young and old mice, and she either treated with an ice-type antibody, or she depleted the CD4 cells with an anti-CD4 antibody. And what she found is, if she depleted the CD4 cells, she got about an 80% loss of the serum IL-10 levels. And so this suggests that CD4 cells are required for this maintenance of this systemic IL-10 increase. And then she did the same thing by depleting Tregs and TFRs, I should say. So both TFRs and Tregs are depleted when she adds DT to FOXP3 DTR mice. And the IL-10 levels actually went through the roof. And so CD4s, but not Tregs, are really critical for this accumulation of IL-10 production in these animals, right? And so that got us thinking, these FOXP3 negative IL-10 positive cells, are they regulatory? Could they be contributing to immune suppression? And so we stepped back and kind of went back and looked in the literature, and we found a few papers that were kind of interesting. So a number of years ago, a paper was published showing that CD4 T cells increase IL-10 with age, and it was correlated with memory cells. I don't think they even discovered most of the cell subsets in 1994, but it was just correlated with memory CD4s. But it's been known for years that high interleukin-10 production is associated with low antibody responses to influenza vaccination in the elderly. And it's actually the interferon gamma IL-10 ratio that's really critical in that regard. But these prior studies really lacked a phenotypic identification of the cellular sources of IL-10, the mechanisms that promote these cells, and really their functional impact in aging. And so for the longest time, we were calling these cells TR1 cells, because that's pretty much what everybody calls a CD4 cell that's FOXP3 negative but makes IL-10. But one of the things we noticed was that these cells really lacked expression of CD49B, which is a canonical marker of TR1 cells. And so that didn't quite fit. We also noticed that they didn't co-produce IL-4 or IL-17. And there was a beautiful paper by Rich Flavel's lab showing that XTH17 cells can actually spontaneously convert to producing IL-10. And we got fate MacBang mice, and we were all excited to show that these are all XTH17s, but that didn't turn out to be the case. Less than 1% were XTH17. We also asked, how many of these were maybe XTregs? So we got FOXP3, Cree, Rosa, lock stop tomato mice, and about 15 to 20% were XTreg, but that frequency didn't really increase with age at all. So it didn't seem like the accumulation was due to a population of XTregs. And then we asked, is the microbiome, are changes in the microbiome driving this whole process? And the answer to that was no, because they accumulate perfectly well in germ-free animals. And actually, this is a daunting experiment, and we're just very fortunate that we have a germ-free facility here, because to keep those mice around in a germ-free environment for 20 months is daunting. So very grateful to our collaborator, Teresa Allengott, for those experiments. But then we noticed that these cells in our array of cytokines that we were testing what they kind of co-produced, we were surprised to find that a fair number of them co-produced IL-21. And you can see here, there's an increase in IL-10 positive cells that co-produce IL-21. And the numbers of cells, the IL-10 positive cells that also produce IL-21 are dramatically increased. And then we gated on the IL-10 positive cells, and we asked, okay, how many of these cells are actually have markers of TFH cells? Because it made sense to do that since they were producing IL-21. And what we found is about half of these cells actually produced, expressed PD-1 and CXCR5. So they had the phenotype of TFH cell. And again, here you can see the increase in IL-10 positive TFH cells is just very dramatic in these old animals. And so because of that, and because IL-6 is important for TFH differentiation, we asked, and the fact that IL-6 is increased with age, we said, does this IL-6 contribute to age-driven TFH accrual? And so we got some IL-6 deficient animals, and both young and old, and we found that the accumulation of TFH cells in general is decreased pretty dramatically in IL-6 knockouts. And then when we looked at TFH10, so we're calling these IL-10 producing Tfh cells, TFH10 cells, and those cells are dramatically decreased with age. In fact, they don't really increase from young to old. So IL-6 is required for this TFH10 accrual. And interestingly, as we'd kind of expect, if these cells are the major sources of IL-10, then serum levels of IL-10 are decreased in these IL-6 knockouts. Right? And that, again, that brings it down to more of a young level. And also, as you'd expect, looking at IL-21 positive cells, of course, those are decreased as well in the IL-6 knockouts that age. And so we wondered if IL-21 contributed to TFH10 accrual. And this is where I really love my friends and colleagues, because I called up a really good friend of mine who I've known for over 20 years, Alan Zajac, and I said, hey, Alan, I knew we had IL-21 knockouts, and I said, do you have any retired breeders or any mice that have just been sitting around and you just haven't sacrificed them yet? And he said, well, actually, we do. And I told him the whole story where we were at, and he said, yeah, that sounds fantastic. I'd love to collaborate. And so Alan collaborated with us on this part of the story. And so in his IL-21 deficient animals, again, we saw this loss of PD-1 CXCR5 defined TFH cells with age. And the TFH10 cells were also substantially lost with age. And again, there doesn't seem to be even an increase at all from young animals. So this accrual is really requires both IL-6 and IL-21 independently. And as you'd expect, again, with the loss of IL-21 and the loss of these TFH cells, systemic levels of IL-10 are decreased. The interesting thing that we found was that systemic levels of IL-6 are increased, as if this IL-10 is acting to kind of suppress this inflammation response, right? So we then wondered, is this something that just happens, it just accrues with age, or if we actually purposely immunize animals, can we see these TFH10 cells that emerge, right? And so what we did is we took influenza NP, the nucleoprotein from influenza, and we just stuck it in alum, and we immunized young and old mice. And the reason we did this is we've got MHC tetramers that we can use to define the T cell response from these animals. And if we look, we can see that we get an increase in the frequency of cells that are binding the IAB tetramer, looking at young mice and aged mice, and then gating on these tetramer positive cells. We then looked at the phenotype because the mice that we immunized were FOXP3 RFP and IL-10 GFP. And we can see that these TFH10 cells really only emerge in the aged animals. And very few of them have FOXP3 positive phenotype. So we, and this just enumerates the total numbers of these cells, so you can find a pretty dramatic increase in these antigen-specific TFH10 cells in the old mice, right? And so then we switched back, and now we used an NP-KLH model. So this is no longer nucleoprotein from influenza, but this is nitrophenol, so that we can track the antigen-specific B cell responses using NP tetramers. So we thought, let's look at what happens in aged mice and what the role might be of IL-10 in controlling the antibody response. And so what we did is we first established a model where we immunize mice with NP-KLH and then look at the NP-specific B cells, shown here with NP tetramer by IgG1. And you can see that the old animals have a significant decrease in responsiveness. So the numbers of germinal center B cells that are NP positive and IgG1 positive are substantially decreased, both the frequency and number. The serum IgG1 levels that are specific for NP are specifically decreased. And so we wondered if IL-10 contributes to this decreased vaccine responsiveness in aged animals. And so what Maha did was to neutralize IL-10 with an antibody against IL-10 receptor, and then she also depleted mice of TFRs and Tregs using diphtheria toxin, just because TFRs have been reported to control vaccine responses in the elderly. And so when she did that, you can see that depletion of IL-10 in that context led to a resurgence of NP-specific B cells. As shown here, serum IgG, NP-specific IgG1 was increased significantly. And depletion, greater than 95% depletion of TFRs and Tregs did not restore that response. So that was really interesting, and we wondered if these TFH10 cells accumulated in aged humans. And Claire Chenier, who's a human immunologist, her lab did these experiments, which are very, very informative. So she had been collecting from our organ procurement organization, which is our organization that provides tissues for donors for tissue transplantation. She had been grabbing spleen cells from people that have had car accidents or other factors that we think would be not very immunosuppressive effects on the spleen cells or the tissues that we get from these people, because we want to have as close to unmanipulated tissues as possible and things that would inadvertently affect immune responsiveness. And the other reason to use tissues is because that's where TFH cells function. They don't function in the blood. They can be seen in the blood, but it's really unclear TFH cells. So to just summarize, we've identified a novel population of IL-10 producing T cells that bear markers of TFH, and these accumulate dramatically with age in mice and humans. Both IL-6 and 21 are required for the accumulation of TFH10 cells. IL-21 is critical for controlling the systemic balance between IL-6 and IL-10. And really, what our data suggests is that these age-related decreases in antibody responses is reversible, right? That's really important and actively suppressed by IL-10. So that has big implications for vaccine responsiveness, right? And so what we think about in these TFH10 cells and aging is that there's both beneficial effects because you counter-regulate the effects of IL-6 and you control how much IL-6 is being produced systemically, but it's also deleterious. So you have reduced efficacy of vaccination as well. And so we're interested in figuring out what these cells do in terms of cancer, atherosclerosis, neurodegenerative disease, osteoarthritis, all of these factors that seem to pop up with age, and IL-10 and inflammation may contribute to. So once again, we go back to that inflammation and frailty slide that I first showed, where we think that IL-21 might be a critical fulcrum between IL-6 and IL-10 in controlling the level of susceptibility you have to age-related diseases versus longevity. And it's really this IL-6, IL-10 yin-yang that's kind of counter-antagonizing itself and setting a balance in aged individuals. It seems to be set by the levels of IL-21. And so this has been a really fun and very collaborative effort, as I said before, between my lab and Claire Chenier's lab. Again, I try to give credit where I ran into the contributions of other people. Teresa for the germ-free animals. Emily and Nathan did a lot of the initial analysis of the single-cell ATAC-seq and single-cell RNA-seq. Sing Sing gave us the FOXP3 fate mapping mice. Sanat helped with the isolation of tissues. Harinder helped us with the NPKLH immunizations. Alex Dent helped us with some TFH questions that we had and is actively collaborating on the project. And this is this international training grant, which funded Christoph Holscher and his student, Iridi, that came to the lab to work. And I acknowledged Alan for his very generous contributions of the IL-21 deficient animals. And so be more than happy to take any questions. Thank you.
Well, thank you very much for this beautiful work. So the first question would be like, how do you think age is promoting the generation of these TFH cells?
Yeah, that's a great question. So, we know that the aged environment produces a lot of IL-6, and we think that this accumulation of IL-6 is a factor that's required for the development of these TFH cells. So if we knock out IL-6, as I showed, the cells pretty much go away. We don't know what types of antigens they're responding to. We were in some ways disappointed to find that the germ-free mice accumulated these cells. But it makes sense now because Alex has seen the accrual of these cells at University of Indiana. Our collaborators in Germany have seen the emergence of these cells in their aged mice. Alan at the University of Alabama has seen these cells in his mice. And I'm nearly certain that none of these institutions do the mice have the same microbiome. So it makes sense that now that I look back at it that the germ-free mice really didn't have much of an impact. But we don't know what the specificities are of these cells. So it's a good question.
Okay, another question from Eugenio Contreras, who's the host of the Open Science Box. He says, congrats on an excellent work, and thank you for accepting my invitation. Does IL-10 signaling is autocrine in TFH10?
So does IL-10 does the IL-10 feedback and then act on TFH10 cells? So I can tell you that Maha, so there is some papers suggesting that if you culture naive T cells and you give them a TCR stimulation, then you add IL-10, that will cause T cells to make IL-10, right? And so there's published literature on that. Maha did that early on, but I don't think she found that IL-10 promoted a TFH phenotype. I think she saw that it enhanced their ability to make IL-10, but it didn't promote a TFH phenotype, if that makes sense.
Okay.
Yep.
So Stephanie Eisenberg, she's asking, well, she says, beautiful story, David. How do you think the TFH10 cells compare to IL-producing Tfh cells observing non-H mice or patients with autoimmunity?
Yeah, that's a great question, Stephanie. We're just starting to look at autoimmune patients. We haven't really seen that much yet. And in the non-aged animals, we haven't seen very many of these cells. And like I said, when we purposely immunized animals and then track the antigen-specific ones, we can't see too many TFH10 cells in that particular context. So I don't know if it's something about the aged environment that's unique that really promotes these cells. I know there's a couple other reports of IL-10 producing TFH cells that came out right when we were in the midst of this work, and beautiful studies, one by Wei Gu in Milwaukee, who showed that in the context of a chronic infection, IL-10 has actually had the opposite effect of what we find, and that is that IL-10 seemed to promote antibody responses in chronic viral infection. And similarly, there was a JEM paper that showed that in tonsils of humans that they could get TFH cells that produced IL-10 that seemed to contribute to allergic antibody responses. So so that was some interesting work. And again, both cases, they described IL-10 as being helping antibody responses. And that actually was one of the first described roles for IL-10 was a B cell stimulatory factor promoting antibody responses. So actually, our stuff is really out of line with that. So we're interested to figure out how that all works.
So I have another question from Jerry Lynn. She says, many IL-10 producing CD4 T cells are also co-expressing interferon gamma. Is that the case for TFH10 cells?
Yes, yeah, a fair number of them will also produce interferon gamma. Yep, we've seen that. They don't co-produce IL-4, at least in the immunization conditions and steady state conditions that we've looked at, or IL-17, but they do, a fair number of them, I'd say probably about 20 to 30% of them co-produce interferon gamma. And an even smaller frequency will make all three cytokines, 10, 21, and interferon gamma.
Okay, wonderful. Well, thank you very much. I think we don't have more questions here. If Alisa, do you want to take over?
Yeah, sure. So thank you everybody for coming. That concludes our seminar on TFH cells. Thank you to the speakers. Thank you, Shane, for talking to us about BCL6, the repressor of repressors, and to Stephanie for sharing your story on TFH independent IgA responses in the guts. And also thank you, David, for giving us insights onto this new TFH10 cells and their roles in aging. And thank you to the audience as well. You guys have had some really great questions, and there's been some really good discussion. And last but not least, thank you to Eugenio for bringing us all here together to talk about our favorite topic, TFH cells. Thanks, everybody.
Thanks for having us.
Yeah, thank you very much.